Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you find these deep dives valuable, please take a moment to like this video and subscribe to our channel. It truly helps support the SANS Science Experience and allows us to keep bringing you these insights. Today, we're embarking on a fascinating deep dive into one of biology's oldest and, well, most persistent mysteries: aging. It's a universal experience for every one of us, and let's be honest, it's the primary risk factor for almost every major human disease, from cancer to neurodegeneration. This isn't just about wrinkles. It's a massive societal challenge. But what actually orchestrates this incredibly complex process at its most fundamental molecular level?
That's the million-dollar question, right? And it's precisely where the dynamic field of epigenetics has really emerged as a crucial player. For a long time, we kind of thought of aging as simple wear and tear, like an old car just breaking down over time. But increasingly, the scientific community is viewing it as something deeper, a profound loss of vital epigenetic information. Imagine your body's instruction manual, the one that tells your cells how to function properly, getting smudged, or maybe even losing entire pages over time. That's sort of the core idea.
Okay, and our mission today then is to unpack a truly groundbreaking new study. It zeroes in on a very specific epigenetic mark: Histone 3 lysine 9 trimethylation. Thankfully, that's abbreviated as H3K9me3.
Phew!
Yes, much easier.
So for years, we've seen this strong correlation, right, between the loss of H3K9me3 and the onset of aging. But this paper asks the critical question: is this loss just a symptom, happening alongside aging, or does it actually cause aging? This is where it gets really compelling, so let's unpack this. Okay, so H3K9e3. Let's try to visualize it simply. Imagine your DNA, your genetic blueprint. It's like a vast library. Our DNA is wound really tightly around these protein spools called histones. And H3K9e3 is essentially a chemical tag, almost like a specific do not disturb sign or maybe a strong lock placed on certain sections of that library. Its main job is to form something called heterochromatin.
Heterochromatin.
Right. Those are the highly condensed, tightly packed regions of our chromosomes. And these regions are absolutely crucial for keeping certain parts of our genome, especially what's sometimes maybe unfairly called junk DNA, silenced and quiet. It's like a genomic bouncer, keeping things orderly.
Right, a bouncer. So if this H3K9e3 mark is like that do not disturb sign, why is it so incredibly important? I mean, what happens if that sign starts to fade or just disappears?
Well, that's the key. Without that sign, parts of our genome that really should remain silent can suddenly become, very active. And that's where the problems start. We're talking specifically about transposable elements.
The jumping genes.
Exactly, often called jumping genes or retrotransposons. These are bits of DNA that, just like their name suggests, can literally jump or move from one spot to another within our genome. Normally, H3K9e3 acts as a guardian, keeping these TEs suppressed, locked down. But when H3K9e3 is lost, these jumping genes are kind of unleashed. They become active, mobile, and this isn't harmless activity. Their movement can cause direct DNA damage. It can activate the innate immune system.
Which tricks your body.
Yeah, it tricks your body into thinking it's under viral attack. And crucially, it can contribute to a whole host of age-related diseases like cancer and autoimmune disorders. This whole concept is really central to what's known as the heterochromatin loss theory of aging.
It's a powerful image, these jumping genes sort of running wild. And you said, historically, this global loss of H3K9e3, it's been seen in lots of species.
In many species, and also in human samples from older individuals or people with premature aging syndromes like progeria.
Okay, but there's always been this huge hurdle showing causation in mammals.
Yeah.
Because if H3K9AV is essential for early development, how do you study its impact on aging later in life? You can't just knock it out from the beginning, right?
Yeah.
The organism wouldn't even develop properly.
Exactly. That's precisely the scientific challenge this paper tackles quite brilliantly, actually. The researchers engineered an ingenious new mouse strain, which they've dubbed TKOC mice.
TKE.
The TKO stands for triple knockout. It means they specifically inactivated the three key enzymes, sub39H1, sub39H, and setDB1, that are responsible for placing that H3K9A3 mark onto the histones.
Right.
So that was their first step towards gaining really precise control over the specific epigenetic switch.
Okay, and here's where it gets really clever, addressing that developmental roadblock you mentioned.
Yeah.
To get around H3K9E3 being essential early on, they made its loss inducible. Is that right?
Yes, exactly. They used a tamoxifen-mediated system. Think of it like a genetic switch that they could flip only in adult mice. This allowed them to turn off H3K9me3 function in mice that had already developed completely normally. It gave them a clean slate to study aging specifically.
Clever.
And just to be sure their genetic switch worked as planned before using whole animals, they did some in vitro validation. They used mouse tail-tip fibroblast skin cells, basically. When these cells were treated with the activating compound, tamoxifen, they saw a dramatic drop in H3K9 and MY3 levels. And crucially, this loss led to decreased cell proliferation. The cells struggled to divide.
Okay.
And they saw a significant increase in cellular senescence markers, things like CDKN1A and IL-6, which indicate stress and inflammation, and also SA-beta-galactosidase activity, that's a classic sign of senescent or zombie cells. So this really confirmed the direct cellular impact of losing H3K9 and F3.
Right. So after confirming their switch worked perfectly in, individual cells, that it could actually slash H3K9me3 levels the moment of truth arrived, what happened then when they flipped that switch, not in a dish, but in a fully developed adult mouse?
The results were, well, quite stark, really provided incredibly compelling evidence. After just 2 rounds of tamoxifen injections, the TKO-Psi mice displayed a severe age appearance, just visibly older. They showed a significant jump in their frailty index. That's a comprehensive way to assess overall health, measuring things like changes in coat condition, increased spinal curvature kyphosis, like a hunchback,
Wow.
and tail stiffening. They genuinely look like they'd aged dramatically, far, far beyond their actual chronological age.
So it wasn't just skin deep, right? What about their actual functional abilities? Did this induced aging affect how they moved, how they lived?
Oh, absolutely. The TKO-C mice showed drastically decreased activity levels, much slower movements, and significantly reduced grip strength. Clear signs of neuromuscular function loss. They also experienced this profound and, importantly, unrecovered reduction in body weight. But maybe the most undeniable finding, the real smoking gun for causality, was the dramatic reduction in their lifespan.
How dramatic.
Their median lifespan was just 30 weeks, which is astonishingly short for a mouse. This is powerful, direct evidence supporting that causal role of H3K9me3 loss in driving aging.
Only 30 weeks? That's incredible. And what about their internal systems? Were there signs of this accelerated aging deep inside their organs too?
Yes, very clear and widespread signs of multi-organ degeneration. It wasn't just one thing failing. They saw signs of anemia, for instance, reduced red and white blood cells, lower hemoglobin, things you often see in aged individuals. Their skin thinned dramatically. They lost essential hypodermal fat. In the small intestine, there were significant structural changes.
Like what?
Things like a reduction in the number and length of the villi. Those are the crucial finger-like bits that absorb nutrients along with crypt deepening. And strikingly, they showed noticeable bone loss, decreased bone volume fraction, thinner cortical bone, really mirroring what you see in normal physiological aging. It was almost as if every major system in the body were shouting, "I'm old," all at once.
Okay, so the outward signs, the organ degeneration, undeniable.
Yeah.
But the real deep dive, as you say, is understanding what was happening inside at that molecular level to drive all this.
Yeah.
How did the researchers actually measure this accelerated aging at the sort of cellular blueprint level?
Right. To measure their biological age more precisely, the researchers turned to epigenetic clocks.
Not actual clocks.
No, not physical clocks. They're sophisticated algorithms that analyze patterns of DNA methylation. Those are other chemical tags on our DNA that change quite predictably with age. They are actually incredibly precise tools now for measuring true biological age, often more accurately than just counting birthdays. And in these TKO-C mice, the clock showed a clear, accelerated epigenetic age, especially in rapidly dividing tissues like the skin, spleen, and small intestine. There was also a global loss of DNA methylation overall, which fits perfectly with H3K9me3's known role in helping maintain DNA methylation patterns, so widespread deregulation at the molecular level.
Okay, and what about the genes themselves, their gene expression patterns? Did they show a signature consistent with, being older?
They absolutely did. The mice displayed what's called a pro-aging transcriptome. That just means the overall pattern of which genes were active or inactive mirrored that of natural aging. They saw a significant upregulation, more activity of genes involved in immune and inflammatory responses across multiple tissues. That suggests a chronic state of inflammation, sometimes called inflammaging. And interestingly, even genes related to multicellular organism development were sort of abnormally switched back on. Conversely, there was a clear downregulation, less activity of genes responsible for normal tissue-specific functions, things like metabolic processes in the liver or ion transport in the kidney. It's a classic hallmark of aged tissues losing their specialized function.
And here's where it all comes back, beautifully in a way, to those jumping genes we talked about right at the start. The very things H3K9me3 is supposed to keep quiet.
Precisely. This is the critical links, the mechanism really clicking into place. Across all 7 tissues they examined in the TK-osic mice, there was a robust, widespread upregulation of transposable elements. Ts were going wild. The dominant families that were upregulated were endogenous retroviruses, ERVs, and long interspersed nuclear elements, or lines. And this uncontrolled derepression, this unleashing of Ts, it directly correlates with the immune activation and the cellular senescence we discussed earlier. It perfectly mirrors observations made in natural aging, too. It really brings that heterochromatin loss theory full circle. It shows how these mobile elements, once unleashed by H3K9me3 loss, directly contribute to the aging phenotype.
Right, connects the dots. So what does this all mean for us then, for the future of aging research? What's the profound so what here?
Well, I think this study provides incredibly strong, direct, and importantly, causal evidence that the loss of this specific bit of epigenetic information, H3K9me3, isn't just happening alongside aging. It's a direct driver of the aging process in mammals. It really elevates the heterochromatin loss theory from being a compelling correlation to, well, a proven causal relationship, at least in this model. And this raises a crucial and maybe hopeful question for future research. If losing this mark drives aging, could restoring it perhaps slow down or maybe even reverse the process?
That's the truly exciting part, isn't it? The potential for intervention. The paper suggests that, right? That targeting these epigenetic modifications.
Yes, it explicitly suggests that interventions targeting these marks may be strategies to restore H3K9me3 levels, and that's something that has actually been seen in some cellular reprogramming studies, or even methods to directly reduce the expression of these troublesome repetitive elements, these TEs, could potentially slow down or even reverse age-related decline. Imagine the possibilities there.
It's pretty staggering to think about.
Indeed. This research undeniably reveals the profound importance of epigenetic regulation in the whole aging process. It really opens up entirely new and I think very promising avenues for developing therapeutic strategies aimed at delaying the onset of age-associated diseases, preserving health span, in our rapidly aging societies worldwide. And importantly, the TK-osic mouse model itself, it's now an invaluable new tool for digging even deeper into these complex, yet incredibly impactful mechanisms.
What an incredible deep dive.
Yeah.
Really showing the causal role of these epigenetic changes in aging. It just highlights how fundamental these molecular mechanisms are to the whole process of getting older. And crucially, how understanding them could lead to potentially transformative breakthroughs in how we approach health and longevity down the line. We really hope you enjoyed exploring this fascinating scientific breakthrough with us today. Remember, if you did find it valuable, please hit that like button and subscribe for more deep dives into the latest, most exciting papers and preprints in science. Thank you for joining us on The Deep Dive, part of the SANS Science Experience.